Okay, so taking off from where we left off. Um, so as I finished with on the previous video, the liver, along with the pancreas, is working towards maintaining homeostasis of blood sugar. And so the connection between the pancreas and the liver is the hormones insulin and glucagon. All right, and so they ultimately have counter effects. And so when we eat a meal, um, our nervous system stimulates the pancreas to secrete insulin. And that's because as we eat our meal, that meal is going to be broken down and cause an increase in blood sugar. So there's an increase in glucose when we eat, and this stimulates release of the pancreas, release of insulin from the pancreas. And so like other hormones, insulin is a hormone and it is a chemical messenger within the body. And so specifically, it has a number of effects which will help to take up glucose from our bloodstream. So as we indicated up above, glucose is going to be absorbed into the bloodstream. This is going to cause a rise, a significant rise in our blood glucose, upon which stimulation of the pancreas to secrete insulin then starts to cause a cascade of metabolic activity that helps to reduce that blood sugar back to normal and homeostatic conditions. So we can sort of see the summary in this box over here on the right-hand side. And that is, the fed state, insulin dominates. And so, you've eaten your meal. And the release of insulin causes this cascade of events. So insulin will cause an increased glucose oxidation, and that makes sense with respect to oxidation, with respect to cellular respiration. It's going to, and here's where the liver comes in, it's going to stimulate glycogen synthesis. So that this insulin is going to cause an increase in the production of proteins that, remember, facilitated transport, the uptake of glucose. And so the liver, along with muscle cells and other cells of your body, but the liver and muscle cells store an enormous amount of glucose as glycogen. And so insulin promotes the production of proteins that facilitate the uptake of glucose. If you remember back from chapter 5, in the transport of glucose is a facilitated transport with channel proteins providing the pathway of glucose into the cells. And so insulin will promote the production of those proteins. Organs like your liver and muscles will take up that glucose and they will store it as glycogen. All right. Now, insulin has other effects, as it indicates in this box over here. Other hormonal effects include fat synthesis. So this is, you want to think of the fed state as being sort of that, the body's getting in nutrients, and so let's build, right? Let's put things together. So we've got fat synthesis, protein synthesis. These metabolic activities are going to be driven by the presence of insulin, all right? And that insulin was driven from the increased blood glucose in our the increased blood glucose levels in our blood. Now, the opposite of this glucose spike is in between meals, our blood sugar starts to drop. And so this is what we would consider our fasted state. So we're not eating 24/7. We have breaks between our meals. And in that, 3 to 5 hour period, hopefully, between meals, your blood sugars are slowly going to be dropping. And we're in a sort of fasted state. And as these blood sugars drop, that leads the pancreas to release glucagon. And glucagon has essentially the opposite effects. Glucagon stimulates rather than taking up glucose and storing it in the form of glycogen, glucagon leads to the breakdown of glycogen and release of glucose into the bloodstream. Thereby restoring blood sugar levels back to normal and homeostatic conditions during the fasted period. So that specifically is what we see here in the box, glycogenolysis, right? And so lysis, remember, split, right? So this is the process of splitting glucose off of that large glycogen molecule, releasing it into the bloodstream. Now, these other two relate to other metabolic processes that all can relate back to the opposite of what's going here. So rather than synthesizing and building up fat molecules and proteins molecules, the opposite, gluconeogenesis, is the conversion of proteins and fats. So the conversion of these, I'm going to rewrite this here so it's more clear, but gluconeogenesis is the conversion of non-carbohydrate molecules, fats, proteins, into sugars. And, again, this kind of relates back to that chapter 6 in which we looked at cellular respiration and the fact that, fats and proteins can be broken down and modified into the products of glycolysis. So if we start with glucose, we're going to go through glycolysis and produce that pyruvate that gets converted into acetyl-CoA. If we start with fats and proteins, we bypass glucose, but we can still generate those compounds which will eventually become acetyl-CoA and enter into Krebs cycle and produce our energy. So, we've got these opposite effects. Insulin, the fed state, let's store, let's build, right? And glucagon, the fasted state, let's break things down, let's keep that blood sugar constant, let's obtain our sugars from other sources, fats and proteins, etc. And lastly here, the effects of glucagon can also stimulate ketogenesis. Now this we could talk about a little bit more in detail when we get into our nutritional aspect of this unit, but essentially this is the conversion of fats into ketones or ketone-like compounds. Or also there's certain proteins that are, there's certain amino acids that are referred to as ketogenic amino acids. So these amino acids can be converted into ketones. And essentially in the fasted state is a result of low blood sugar and low glucose. And so the body is looking to provide energy for working cells from other sources. And so ketogenesis is the process of breaking these fats and amino acids into other non-sugar compounds referred to as ketones, just another organic molecule that the body can metabolize for energy. All right, so these are two very important hormones to understand. And if you have questions about that, make sure you bring them to class. The last three are hormones specifically related to the digestive process and the breaking down of food and how it stimulates the action of all these things that we've talked about as food passes from the esophagus into the stomach, stomach into the small intestine, and so on. So starting in the stomach, gastrin is the hormone that is released from the stomach. So you can see it here. It's released, the cause of its release is due to peptides, so small amino acid chains and amino acids themselves that are present in the gastric lumen. So basically, the cavity of your stomach. And the effects of this is that gastrin stimulates gastric secretions. And so, of course, gastric juice. So it's stimulating the stomach and this lining, the cells of the stomach, chief cells, parietal cells. It's stimulating them to fire up their production of important substances for the digestion of amino acid proteins. So in secretion of gastric juice, in that gastric juice, you have pepsin, the enzyme to break down proteins, along with hydrochloric acid, which is required in terms of the metabolic pathway and breaking down proteins and keeping that pH at a low level at which pepsin acts. All right. Then secondly, we have cholecystokinin, which is abbreviated by CCK, sorry, and essentially the presence of both fats, or I should say fatty acids and amino acids in the small intestine stimulates the secretion of CCK. So the small intestine releases the hormone CCK, and this has a double effect. One is it stimulates action and contraction of the gallbladder. So contraction and emptying of the gallbladder, and that makes sense if the gallbladder helps emulsify fats and helps with digestion. So it makes sense that the presence of fatty acids in the small intestine would stimulate the gallbladder to contract and release that bile to help with the emulsification of those fats. And it has a secondary effect coming back on the pancreas and stimulating action of the pancreas to secrete pancreatic juice. And again, here we need the enzymes coming from the pancreas like lipase that will aid in fat digestion as well, right? So okay, last hormone is secretin, and secretin is also released from the small intestine. And what causes its release is the increased acidity in the pH in the lumen of the small intestine. And of course, that happens as the stomach, as the pyloric sphincter of the stomach opens and releases that acidic chyme into the small intestine. That acidic chyme is going to stimulate the small intestine to release secretin, and secretin has an effect on the pancreas, again, to release that pancreatic juice, right? Which, again, the pancreatic juice contains water to aid in digestion and lubrication. It contains the enzymes like trypsin for the breakdown of proteins, lipase for the breakdown of fats, but also, remember we've talked about bicarbonate. And so this bicarbonate is effective at maintaining the pH, acting as a buffer, right? Maintaining the pH of the small intestine at around 7.8 so that these enzymes, trypsin, lipase, etc., can function properly. And of course, secretin stimulates the secretion of water and bicarbonate from the pancreatic and bile ducts. So, and that's it. All right, enjoy the rest of your weekend and good luck with all the studying.